Hello friends, as we all know, we are starting DNA replication from our previous class. We are going to deal with the next part that we left yesterday. We discussed the replication enzymes on our last class. So we are dealing with the process today in detail. So I'm not writing everything that we discussed yesterday. Those were the enzymes. So this process of replication meant that we are going to double the number of DNA so that when the cell divides, both the two new cells get the replicate copy of DNA. So for instance, suppose this is our DNA that we need to double. So the first enzyme we mentioned yesterday was helicase, which was supposed to sit on a site where replication originates. The site where replication will originate, we also say this term as ori-C site. So this helicase enzyme is going to open up the DNA or open the helix to some extent initially. The helicase enzyme is followed by the enzyme primase, as mentioned earlier yesterday, that is going to place the primer in a very sophisticated manner. The point is that the DNA molecule are always having a direction. That is, it is always, the two strands of the DNA molecule are always anti-parallel in fashion. And when the primase is going to make the primer for them, it is also following the same rule that the primer is going to sit in an anti-parallel fashion. As I have already mentioned the function of primer in our last class, I am not going to discuss those things again here. So, when the primer is being set by the primase enzyme, the next enzyme coming to play is polymerase. The most predominantly coming enzyme that is for our second year syllabus is the DNA polymerase 3. So what it does, it is going to bring one nucleotide at a time and is going to make them sit one after another. Okay? So there is another termination that when these three enzymes comes once after another, this helicase and primase, when it comes to sit for the first instance, we call as primosome. And when polymerase enzyme comes to play, these all three enzymes together are known as replisome. So at this point, this is like a fork. When this opens up, this is like a fork. We have two parent strands. One we are going to be calling as leading, and the other one is known as lagging. So what's the difference? Point is that when primase is going to make the primer in one strand, it can synthesize the strand in this direction up till the point helicase open the DNA strands. But if you see the primase for the other strand, it cannot make the primer sit here. Even if it sits, there is no significance. And because this is the 5' of the DNA strand and the primer has to be 3'. If this is 3', this will be 5'. As we all know that the DNA replication can occur only in the direction of 5' to 3'. So here, if DNA replication has to occur, it needs to proceed in this direction. But there is no parent strand in this position, so it cannot form any complementary base pair. So there is no significance of making sit this primer at this site. Hence, the theory changes for this strand. What it does, the primase enzyme is made to sit the primer at this site. So this is 5', this is 3 prime. So when the polymerase enzyme will find that free 3 prime hydroxyl at this site, what it will do? It will proceed in this direction till the end of the parent strand. What will happen in the next step? As soon as the helicase enzyme will move forward in this direction, a more stretch of DNA will be opened up for the process of replication. So what was the previous scenario? We are going to draw that first. See, this was our primer having 5 prime, 3 prime. Till this point, it was being synthesized till here. The last nucleotide that was being added at this position, this one, will having a 3 prime free hydroxyl at this side. Hence, when polymerase will find the 3 prime hydroxyl here, it will again continuously synthesizing the strand up till the point where it will find the helicase enzyme. But the scenery is not here, same here, because till this point the primer was here, it was 5 prime, it was 3 prime, and it was being synthesized in this direction. If polymerase is going to find this site, it cannot synthesize in this direction. As we all know, replication can occur only in the direction of 5 prime to 3 prime. So here it cannot synthesize. Why? Because the polymerase will not find the free 3 prime hydroxyl at this site. And this 3 prime is not free, it is bound by the next nucleotide already. So what primase is going to do? It is going to bring one primer here again, which will having a 3 free prime hydroxyl once again, and we will have another stretch of newly synthesized DNA in this direction. Hence, I'm just rubbing just for your convenience, but there is no such gap. The point is that in this strand, the DNA replication will be continuous as soon as this helicase will move this direction, because it will always find the free 3 prime hydroxyl end every time. But when in this strand new primer has to be synthesized, there will be some small fragments will be added every time the helicase is opening the parent DNA strand in this direction. So these small fragments, we call them as very important term for any exam, that is okazaki fragments. So these okazaki fragments are always shorter than the strand that is being synthesized in the leading strand. So this one where replication is continuously occurring, we call this is the leading strand, and this one becomes the lagging strand where okazaki fragments comes into play. Another very important point is that DNA replication was semi-discontinuous. And another property was semi-conservative. So to discuss semi-conservative, we will require another experiment, so we are not going to discuss in this video. We are dealing with semi-discontinuous nature here. What semi-discontinuous you see? Two strands, one strand, when it opens up, it is always in a continuous fashion. So it is 50% continuous. For other strand, it is not continuous. What it is breaking the fragments every time the helicase opens the parent DNA. That is why discontinuous is how much? 50%. That is why DNA replication is called as it is a semi-discontinuous process. But what is semi-conservative? It's a long experiment, but we are going to discuss in the next video. Another enzyme that was playing in this scenario was the topoisomerase. See the point. I have already mentioned in our last class that when helicase is opening the DNA strand in this direction, there will be more coiling in this side. You imagine the example of flexible wire. When you try to open it, there will be more coiling in the other side where it is not yet open. So that we call as DNA supercoiling. So to remove that supercoiling so that helicase can freely move in this direction, this was the enzyme topoisomerase 1 or topoisomerase 2. What one was doing when suppose these two were tangled more, topoisomerase 1 is going to cut one strand, and swivel this two end on the other side. That means, just imagine, this is a complex structure. So if you have to loosen this structure, what will do? It will cut at one side and join on the other side, so that one tangle is at least cleared once at a time. So this is what going to be done by topoisomerase 1, and the topoisomerase 2 is similar, that it cuts both the strands, release the tension, and again joins it by the enzyme topoisomerase. The other enzyme, that is ligase, is going to join the DNA strand when there is a breakup. Suppose this was a DNA strand and this was not a DNA. This was primer made of RNA. So this RNA was being replaced by DNA polymerase 1 enzyme, and when DNA polymerase 1 is applying the DNA strand here, there will be a gap between these two, and this will be sealed by the enzyme DNA ligase. So this is all about the process of DNA replication. So in our next video, we are going to deal with more videos regarding DNA and RNA, regarding the genetic syllabus we are going to deal with. Thank you.